Good afternoon. Today I will be presenting to you my individual project number 2 in Biochem 316 on nuclease. Here is a structure of nuclease. I will come back to this in just a moment. First, we need to learn what is nuclease. Nuclease is an enzyme that goes into DNA and cleaves the chains of nucleotides in nucleic acid into smaller units. That is what we see here happening in the center of these DNAs. Once it's complete, in the middle is when it's starting to go in the nucleo, and on the last one is when it is separating it into smaller pieces. A nuclease is an enzyme that's capable of cleaving the phosphodiester bonds between nucleotides and nucleic acids, as we saw in the illustration prior. So this enzyme also has an effect on our digestion, the environment within our body, and the environment outside of our body. So we'll be going over a few things that enzymes do in general, as well as our specific focus. In all of living organisms, there are essential machinery for many aspects of DNA repair. Our enzyme plays a big role in that. When there are defects in certain nucleuses, it can change the cause of genetic instability or immunodeficiency, so causing things to be born with an immunodeficiency, and instabilities can cause disease, growth, or deformities. Nucleases are also extensively used in molecular cloning. The pH at which an enzyme functions optimally is called the pH optimum. Their optimum is making a solution more basic or making it more acidic in order to decrease the rate of the reaction that the body is needing at that specific time. So here we've got a lot of words, but I will break it down simply for us. The pH changes that we just mentioned, where it's able to make it more basic or more acidic. These changes are made in order to change ionization. This ionization of an amino acid R group in the protein, as well as the extent to which they are able to bond to a hydrogen bond, can affect and will either enhance or reduce how things are being made within the DNA. They will cause an enzyme to lose its biological active configuration, and it can become denatured. When something a little less drastic is going to happen and denaturation isn't going on, but still big changes are happening, changes in the R group of an enzyme active site can also destroy the ability to form the enzyme substrate complex. So for an example here, we have a quote. I've separated the paragraph into 2 bullets to make it a little bit easier on the eyes. But to kind of paraphrase it, the pH in the stomach is approximately at a level 2. So as a result of that level 2, the secretion of hydrochloric acid, also known as HCl, by our cells in the stomach lining when breaking things down, the proteolytic digestive enzyme pepsin must effectively degrade proteins at this extreme pH, which is hard for it to do considering it's at a different pH level. So in this case, pepsin, the enzyme, has involved an amino acid sequence that can maintain a stable tertiary structure at a level of pH 2, and is most effective in the hydrolysis of peptides that have been denatured by a very low pH. Thus, pepsin has a pH optimum of 2. So as we learned earlier, the optimum means it's when the body is kind of going to the level at which it needs to in order to perform its specific job. So here, although we're speaking of the enzyme pepsin, it's just to give a further example of what all enzymes support and do. When the stomach needs an enzyme, the enzyme has to configure to the stomach and to its pH level in order to be able to function properly. It's pretty awesome. So then we're going to learn a bit about the temperature optimum. The temperature optimum is where the enzyme is functioning optimally at the rate of reaction that is maximal. So above the temperature optimum, increasing temperature begins to increase the vibrational energy of bonds within an enzyme, and this is going to be important for the separation or the bonding of enzymes and their ability to do so or lack thereof. So eventually, so many bonds and weak interactions are disrupted that the enzyme becomes denatured and the reaction stops, and that causes a whole string of reactions, negative reactions from that. So what is its use in medicine? Physicians can use many enzymes. They have found many enzymes available to treat heart attack patients, They have found some to help with DNA technology, which has provided science with more clot-dissolving enzymes. They have found some that have elevated blood serum concentrations of an enzyme, amylase, and lipase, which are indications of pancreatitis, which is a huge problem within the United States, and inflammation of the pancreas. So liver disease such as cirrhosis and hepatitis, which is widespread over the United States and some of the world. So having an enzyme that can help lipase and amylase in these specific cases is a huge finding. Enzymes in general also can increase concentration following a heart attack, but the physician can differentiate between these two conditions by considering the relative increase in two enzymes. So all the enzymes are going to play a big role in helping not only the patient, but the specific issue going on. They all change levels, just like we heard the acid-base level, basic levels, temperature. Our body's constantly changing to go to where it needs to go in order to heal or to be at homeostasis. So what this is saying is when enzymes are increasing, the doctors are able to tell the difference between which ones are needed and which ones are not in specific conditions. Enzymes are also used as analytical reagents in the clinical laboratory, owing to their specificity. Pardon me on that pronunciation, I said that incorrectly. Enzyme replacement therapy can also be used in treatment of certain diseases, as we've heard, and they have reviewed recent articles, and DNA and RNA include illustrations, reactions. Pardon me, my computer just jumped. They also include one or more drawings, an illustration of reactions, and a summary of the article and its importance. And so what that means is they've found enzymes across the board that have been able to aid, including our specific enzyme, in DNA and RNA defects, in heart attacks, as we learned, in pancreatitis, and inflammation of the liver. So these are all huge findings, and this specific enzyme has done wonders in helping the progression of DNA, even some with DNA. Pardon me, I think my computer cut off. I do apologize, that's been happening frequently. So it can even help in DNA replication, which is something that people have been trying to learn about for a very long time, and I think that we are continuing to get closer and closer, but we've now learned it's not just about the DNA and the RNA. Enzymes have a huge part in playing all of the roles in all diseases and all diagnoses that we're trying to help, fix, and maintain. And then here are my sources. Thank you.